Morning students, now coming to another topic, plastids. These are also a type of cell organelles responsible for photosynthesis, that is assimilation of carbohydrates. So these plastids were first discovered by scientist Schimper. Discovered by scientist Schimper. Generally, these plastids are present in all plants, and also present in euglenoids, plants and euglenoids. So, presence of plastids is a characteristic feature of all plants and also euglenoids. Were discovered by scientist Schimper. Okay. So these plastids they contain different kinds of pigments. They contain different kinds of pigments, and which are responsible for trapping the radiant energy, light energy. Then it is converted into chemical energy. They play very significant role in photosynthetic process, that is assimilation of carbohydrates, by replacing the light energy, water and in the presence of carbon dioxide. So these plastids they contain different kinds of pigments, and they are responsible for trapping the radiant energy and converting into the chemical energy. They play very significant role in a photosynthetic process. Depending upon the type of depending upon the type of pigments, these depending upon the type of pigments present in these plastids are divided into three types. This is the classification of pigments, or this is the classification of plastids depending upon the type of pigments. One type of plastids, they are called chloroplastids. These plastids they contain chlorophyll A. Generally, these plastids they contain chlorophyll A, and they are a type of green colored plastids. So, green colored plastids are called chloroplastids. Mainly, they contain chlorophyll A molecules. Because of this reason, these chloroplastids are they are green colored. Similarly, second type of plastids are they are called chromoplastids. These chromoplastids they contain different different colored pigments. For example, they contain carotenoids. What are carotenoids? Carotenoids means keratin and xanthophyll. Actually, these keratins are orange or red colored pigments. At the same time, these xanthophylls are yellow colored pigments. So the plastids which contain different kinds of pigments, like these keratinoids. Keratinoids are of two kinds. One is keratins, second one is xanthophylls. Keratins are usually red or orange colored pigments. Similarly, xanthophylls are yellow colored pigments. So these chromoplastids they contain different colored pigments like xanthophylls and keratins. These pigments they are present in different parts and they imparts different colors to the plant parts. Especially they are present in petals. Because of this reason, these petals they exhibit different colors and different species of plants. These chromoplastids they play very significant role in attraction of insects. Attraction of insects, which help in the cross-pollination process. Anyhow, green colored plastids are called chloroplastids. Generally, these chloroplastids are present in mesophyll cells. Okay, they are generally present in mesophyll cells. At the same time, the plastids which contain different kinds of colored pigments, like keratins and janths, they are called chromoplastids. They also provide different colors to the different parts of the plant body. Especially, they are concentrated in petals, and petals are in different colors in different species of plants, so that these chromoplastids they play very significant role in attraction of insects, especially in petals. And also these pigments they also absorb the light energy. At the same time, the third type of pigments they are called leucoplastids. Leucoplastids. These leucoplastids they store mean colorless. Leucoplastids are colorless pigments. Colorless pigments. But what is the importance of these leucoplastids? Colorless pigments are called leucoplastids. These colorless pigments they store food material. They play very significant role in storage of food material. They store different kinds of food materials. At the same time, some plastics, some leucoplastids, they store carbohydrates. Some store proteins. Some store oils and fats. Depending upon the type of food material stored inside this leucoplastids are divided into three types. Depending upon the type of food material present inside this leucoplastids are divided into total three types. The first type of leucoplastids they are called amyloflask. Amyloflask. First type of leucoplastids are amyloblasts. These amyloblasts they store carbohydrates. They store carbohydrates. Generally, they are present in potato and tubers. So, these amyloblasts they store some amount of carbohydrates. Generally, they are present in potato and other root tubers, etc. At the same time, second type of leucoplastids they are called alleroplastids. Alluran means proteins. Okay, these store some amount of proteins. Generally, this alluroplasts are present in pulses, grams, red gram, green gram, black gram, Bengal gram, like that. These alluroplasts they store some amount of proteins and generally they are present in pulses. At the same time, third type of plastids, leucoplastids, they are called elaioplasts. They are called elaioplasts. Generally, they store fats and oils. Fats and oils. Best examples, resinous is one of the best examples. Castor, resinous, comments, castor. So, These leucoplastids are colorless plastids and are present generally in the plant pots which are present in deep in the soil where the sunlight is not focused. And these leucoplastids, they play very significant role in the storage of food material. Depending upon the type of food materials, these leucoplastids are also divided into three categories. One is amyloplas, carbohydrates storing leucoplasts, and protein storing leucoplasts are called alleroplasts, and fats and oil storing leucoplasts are called elaioplasts. Example for amyloplas, potato and root tubers, and for alleroplasts, pulses, and examples for elaioplasts, that is fats and oils. Students, now coming to the structure of chloroplast. Okay, structure of chloroplast. Students, chloroplast is a double membrane bound structure. Very important. Double membrane bound structure. Chloroplast is a double membrane bound structure. Double membrane bound structure. Generally, these chloroplasts, the number of chloroplasts per cell, it is vary. It is vary. What is the number of chloroplasts per cell? Generally, each chloroplast, it is missing about 2 to 4. Okay, 2 to 4 micrometers width and 5 to 10 micrometers length. 2 to 4 micrometers width and 5 to 10 micrometers length. At the same time, the number of chloroplasts per cell is also varying. In case of mesophyll cells, the mesophyll cells of leaves is represented by 20 to 40 per cell. 20 to 40 chloroplasts are present in each and every cell of mesophyll cell in leaves. But in some members, that is in case of chlamydomonas. So actually this chlamydomonas, it is one of the unicellular green alga. This unicellular green alga, it contains only one chloroplast inside the cell. Only one chloroplast inside the cell. So chloroplast is a double membrane bound structure, and generally 20 to 40 chloroplasts are present in each and every cell of mesophyll leaves, but only one is present in chlamydomonas. This is one of the unicellular green alga. At the same time, what about the size of this chloroplast? 2 to 4 micrometers width, 5 to 10 micrometers length. That is the size of chloroplast. Similarly, the shape of chloroplast, shape is also varying. Shape is also varying. In some members, they are discoid. In some members, they are lens shaped. But in some members, they are oval shaped. But in some members, they are spherical shaped. But in some members, they are ribbon shaped. Example, spirogyra. Ribbon shaped spirogyra. So, size and actually the shape of chloroplast is varying. In some members they may be discoid, in some members may be lens shaped, but in some members oval, in some members spherical. They are also ribbon shaped in some algal members. like spirocheta. In this way, every chloroplast it is bounded by an envelope which is differentiated into outer membrane and inner membrane. There are two membranes in this envelope. It is a double membrane bound structure which is differentiated into outer membrane and inner membrane. Actually, this inner membrane is less permeable. It is less permeable. The space present between the two membranes, this is called periplastidial space. Periplastidial space. So there is envelope surrounding this chloroplast and is differentiated into outer membrane and inner membrane. The small gap present between outer and inner membranes, that is called periplastidial space. Similarly, the inner membrane it is less permeable. At the same time, the entire inner space of chloroplast, this is called stroma. This is called stroma. Okay. So, inner space is called stroma. Inside this stroma, number of membrane bound structures. Number of membrane bound structures. They run from one pole to the next pole continuously. So, this stroma is represented by number of membrane bound structures which are run from one pole to the next pole parallelly. Such type of membrane bound structures run from one pole to the next parallelly. Such membrane bound structures are called thylakoids. They are called thylakoids. Okay, these thylakoids are membrane bound structures. But in certain locations, these thylakoids may be individual, but in some members they are present in groups. So, if the thylakoids are present individually inside the stroma, such thylakoids are called stroma thylakoids. They are called stroma thylakoids. At the same time, in certain locations, all a group of thylakoids are arranged just like the collection of coins. They are arranged just like the collection of coins. They are called grana thylakoids. They are called grana thylakoids. What is grana? Grana means in certain locations, thylakoids are arranged one above the other, leading to the formation of groups. For example, this is individual thylakoid. They are individual thylakoids. But in some certain places, thylakoids are arranged one above the another. Such group of thylakoids is considered as this is called grana. Group of thylakoids is called grana. Individual thylakoids are called stroma thylakoids. The thylakoids which are present in grana are called grana thylakoids. Depending upon the nature of thylakoids inside the stroma, thylakoids are divided into stroma thylakoids means individual thylakoids and grana thylakoids means present in groups. In this way, several membrane bound structures called thylakoids, they run parallelly from one pole to the next pole continuously through the stroma. They are called thylakoids. Maybe individual stroma thylakoids, maybe arranged in groups, grana thylakoids. And all these thylakoids, very important. And all these thylakoids, they are interconnected by membranes. All these thylakoids are interconnected. They are interconnected by some membranes. These membranes are called stroma lamellae. Stroma lamellae. very important. These membranes that connect the thylakoids of adjacent thylakoids. All the adjacent thylakoids are interconnected by some membranes. Such membranes that connect the adjacent thylakoids, they are called stroma lamellae. They are called stroma lamellae. So, these thylakoids, they contain number of pigments. Thylakoids, they contain number of pigments which are responsible for trapping radiant energy. So, these pigments are present in thylakoids. So, light phase of photosynthesis, it takes place in the thylakoids or grana. Grana means group of thylakoids. So, these thylakoids, they contain different kinds of light absorbing pigments. Due to presence of pigments inside the thylakoids or grana, grana is responsible for light phase of photosynthesis. Light phase of photosynthesis takes place in grana or it takes place in thylakoids. In this way, for example, this is a thylakoid and this is stroma lamellae. Every thylakoid, it is represented by a narrow empty space in its center. Such empty space present in the center of thylakoid, this is called lumen. Very important. This is called lumen. What is lumen? The inner space of the thylakoid. Thylakoid is internally represented by a small, narrow, empty space. Such a small, narrow, empty space present in thylakoid is called lumen. At the same time, this stroma, okay, stroma is responsible for dark phase of photosynthesis. Dark phase of photosynthesis. What happened in dark phase of photosynthesis? CO2 is converted into carbohydrates. In light phase pigments, they absorb light energy, then it is converted into chemical energy. By using this chemical energy, in dark phase, CO2 is reduced into carbohydrates in dark phase. So, dark phase, it takes place in a stroma of chloroplast. At the same time, this stroma is also contain number of enzymes responsible for dark phase of photosynthesis, and also it contain 70S ribosomes, and also it contain RNA, and also contain double-stranded naked circular DNA, just like chloroplast. Very important. Not only in chloroplast, but also in chloroplast. Not only in mitochondria, but also in chloroplast. This stroma is represented by number of enzymes responsible for dark phase of photosynthesis, 70S ribosomes, RNA, double-stranded naked self-replicating DNA. Due to presence of self-replicating DNA, this is also called semi-autonomous cell organelles. Semi-autonomous cell organelles. Both mitochondrion and chloroplast, they contain 70S ribosomes and RNA, enzymes, and double-stranded circular DNA. These characters are common for both chloroplasts and mitochondria. So that both chloroplasts and mitochondria, both are double membrane bound structures, and both are also called semi-autonomous cell organelles, and also called cell within the cell, and also called semi-autonomous cell organelles due to presence of double-stranded naked circular DNA. Anyhow, both chloroplasts and mitochondria, they constitute semi-autonomous cell organelles. At the same time, they are also called prokaryotic cells. within the eukaryotic cells and also called cell within the cell. So, thyroids are granule responsible for light phase of photosynthesis and stoma is responsible for dark phase of photosynthesis. Anyhow, these chloroplast is responsible for the synthesis of carbohydrates.